lecture given by Dr. Jonathan Marchant. So Jonathan is a Marcus Professor and Chair of Cell Biology, Neurobiology, and Anatomy at the Medical College of Wisconsin. So for the people who are not so familiar with our geography, if you can imagine, this is a Michigan lake. Jonathan is here. I'm here. Or maybe Jonathan a little bit here, right? Okay, so that means if I want to visit Jonathan, in principle, I can swim across the Michigan lake. Jonathan graduated from the University of Cambridge and did postdoc training at UC Irvine. Since his graduate school, he has devoted his full effort to study calcium signaling. And today he's going to talk about their very interesting work on the mechanism, how PZQ, a drug to treat parasitic flatworm, act with worm trip channels and cause a large calcium entry into the worm and cause worm paralysis. So for all the audience, please type your questions in the chat box during the presentation, and I will read the questions to Jonathan to answer for the Q&A session. So Jonathan, all yours now. Very look forward to your talk.
Great, thanks very much. Welcome everyone. It's good to see everyone virtually. Thanks for attending today. Appreciate the introduction and thank the organizers for all the efforts putting together this great talk series. So today I want to talk about calcium signaling and neglected tropical diseases, and I think this is an area where, as a field, we're somewhat underrepresented, and it's a shame because I think it's an area where we can make a big impact. Many of the important drugs that are used to treat neglected tropical diseases target excitable cells, so that should be well within our calcium signaling wheelhouse. But even more generally, as academic researchers, given that there's so little big pharma present in this area, despite an urgent need for developing new drugs, I think it's an area where we can and we should be contributing. And hopefully that's one of the take-home messages from my talk today. So my game plan for the next, I'm just going to talk for 30 minutes. I'm going to try and cover 4 areas. I'm going to start with a general introduction about neglected tropical diseases, and our lab is interested in diseases caused by parasitic flatworms. I'm going to talk about this drug called praziquantel, which is probably the most important drug that you've never heard of. It's a drug that has, for a long period of time, been known to disrupt calcium homeostasis, but no target for it has ever been found. I'm going to talk about our lab's recent data where we think we've finally nailed down what that target is, and it appears to be a parasitic worm trip channel that's activated by praziquantel. So I think in retrospect, praziquantel was probably the first ever clinically approved trip channel ligand, but we haven't realized that for 40 or so years. And then this is sort of an informal talk and get together, so I thought I'd say a few words about what happens next and where we'd like to go with some of these projects. And overall, I think our lab's quite excited about this work because I think we're on the cusp of potentially making a difference here. So neglected tropical diseases are diseases of neglected people. They represent 20 or more conditions, diseases that are caused by bacteria, viruses, worms as well, that affect over 1 billion people worldwide. And if I toggle between the next 2 maps, you can see that they look pretty similar. One of these maps is where neglected tropical diseases exist. The other map is GDP per capita, so income. So these are truly diseases of neglect. They're diseases that blind, so river blindness, onchocerciasis is a parasitic flatworm disease. The diseases that disfigure, lymphatic filariasis is another example, commonly known as elephantiasis, a disease that also disfigures, but there are also diseases that disable. So what you can see here are kids holding up tubes of their urine, and you can clearly see the blood that's in there. And that blood is caused because they're infected with parasitic schistosomes, blood flukes. So these diseases affect their health, their ability to go to school, and therefore ultimately entering the workforce and economically contributing. I've got one last slide to talk about impact, and it's a public health slide, so it's not kind of the slide we normally look at in sciencey talks. On the y-axis is additional school years, children's school years gained per $100 spent, and on the x-axis is on the basis of different interventions. So if you give kids free uniforms, have pride in their school, they're more likely to turn up. Merit scholarships also seem to work. They're more effective than sort of giving cash for attendance allowance type things, or just giving kids free cash. So here's the effect of deworming, and you can see really quite a jarring return on investment. That this is really a challenge, an urgent healthcare challenge, and one of health equity. That investing just an extra $100 translates into a considerable gain in education. So some of the culprits for these diseases are on this slide: tapeworms, liver flukes, and blood flukes, which I'm going to talk quite a lot about today. Tapeworms are probably the clinically least important. I don't want to belittle them. However, there are some diseases. For instance, one of the major presentations with epilepsy in Central America is a disease called neurocysticercosis, which is where tapeworm eggs are present in the CNS and they start to cause epileptic seizures. Liver flukes are a major public health problem in Southeast Asia, and you get infected by eating raw or undercooked fish that contain infected larvae. But there are also diseases of livestock, and actually some of these are on the rise even in developed countries. So in the UK, for instance, Fasciola is a growing problem being driven by drug resistance and also global warming. But I'm going to spend most time today talking about blood flukes, so parasitic schistosomes, because they're the most clinically significant. There are 3 species of schistosomes that infect over 250 million people worldwide, and you probably know of schistosomiasis, of sort of snail fever, bilharzia. Sort of this infective stage comes out of the worm, these highly active cercaria that seek out in water people to infect. They're attracted to lipids on skin. And then the sort of a really fascinating sort of journey of the organism in terms of its physiological challenges. The cercaria have to burrow through the surface of the skin, through the epidermis, somehow find a blood vessel. They're then in the circulatory system where they grow and mature. The worms, the male and female worms, become adults. They pair up, they mate in a particular region of blood vessels in your body, so quite commonly the mesenteric vessels. So what you can see in this picture is a male and female worm. They are prolific egg layers. They deposit hundreds of eggs, and it's not really the worms that cause the disease, but it's the inflammatory reaction to the eggs that are a problem. So the worms themselves are masters of immune evasion, so they can live in your body and be undetected for the immune system for decades. It's the eggs that sort of initiate the granulomatous reactions that cause fibrosis, portal hypertension, and the characteristic hepatosplenomegaly, which is associated with schisto. Okay, so all these diseases caused by parasitic flatworms are treated by this antiparasitic drug called praziquantel. So clearly you can see what an important drug this is for keeping these diseases under control. There's one exception to this, and I am going to come back to this a little bit later, but the liver fluke Fasciola is well known to be refractory to praziquantel, and that's been a mystery for a while. But most of the other parasitic flatworms can sort of be treated using praziquantel. Let's just say a little bit about this drug. It's a very simple drug. It's also an old drug. It was discovered by Bayer and Merck back in the 70s. It's widely used. 100 million people get prescribed praziquantel each year. Thankfully, it's very cheap. It only costs 20 cents to treat a child who is infected. And that has enabled the sort of intentional development of mass drug administration campaigns, sort of Gates Foundation type investments to try and eliminate schisto in areas where it's endemic. It's a pretty effective drug, but it's by no means perfect. It has to be given in quite large doses, and you can sort of see on the slide sort of quite a big tablet. Half the dose that we prescribe is actually ineffective because the drug is a racemic mixture. There are two enantiomers here. It's just the R praziquantel, the eutamer, that actually is effective against worms. The S praziquantel does very little at clinical doses, but it does give the drug quite a bitter taste. So in terms of field medicine, getting kids to swallow a large tablet that doesn't taste great is a key problem. It's less effective against juvenile worms, so quite often it doesn't cure infections. And of course, the problem I introduced at the start, we don't really know how this drug works. That no target has been identified. So overall, I mean, I think this is quite an important problem to solve. It's the one drug that treats all these different parasitic diseases. We're not sure how it works. We're prescribing it to millions of people, probably in subcurative doses, with no really other drugs in the pipeline because of the lack of pharmaceutical interest. So the specter of resistance does loom large, which if it were to become established would be devastating. What we do know is that this is a calcium signaling problem. So we've known for a long period of time that if you add praziquantel to schistosome worms, you can immediately see what happens. You get a rapid contraction, a rapid sustained contraction of the worms. And this is, you can see from the tension, from a very old measurement here, it's a sustained muscle contraction. And I think the calcium influx is so intense, it actually damages the worms. The surface of the worms, called the tegument, starts to vascularize, and that sort of allows antibodies to recognize the worm and contribute to their elimination. It's potent, so praziquantel that causes this effect at nanomolar concentrations. It's also quite stereoselective, so at the same concentration, if you use the distamer S praziquantel, the worms don't really contract. So we've known for quite a long time these sort of characteristics of how the drug may be working: nanomolar potency, stereoselectivity, sustained muscle contraction. So we've been trying to figure out the problem about how praziquantel worked for a while, and maybe in our usual fashion, we didn't do this very efficiently. Rather than taking the direct route to the top of the mountain, we've sort of had a long winding road. Parasites are kind of difficult to work with. Functional genomics is not well established. It's difficult to maintain the entire life cycle. For those that know our work, we spent some time sort of working with planarian flatworms, their cuter free-living relatives, and that enabled some progress. But really, and somewhat counterintuitively, but when you get to the top of the mountain, somewhat obvious in the past, the key breakthrough for us was really working with humans. And we got kind of a bit of grief about this from reviewers and grant submissions, with the obvious critique about why the hell do you care about low affinity interactions of an antiparasitic drug in a host that's not supposed to be sensitive to that drug. And that was valid, but for us, we sort of stuck at it and trying to find, because things are much easier to do in human systems, doing screens to find targets was actually quite illuminating for us. So by taking a step back to actually working in humans over the course of several years, we were able to show that the R-praziquantel enantiomer, and this was work by John Chan when he was in the lab, is actually a stereoselective activator of the 5-HT2B receptor, a GQ-coupled receptor. And then Gihan, who talked earlier in the series this year, as well as Alexandra Babe's lab, were able to show that S-praziquantel was a low affinity activator of TRPM8, which may contribute to the bitter tasting target of that drug. And although these are sort of low affinity interactions, what they did give us was sort of a molecular toehold on doing target-based screens back in the flatworm. We know that although there are lots of GPCRs and lots of ion channels in the flatworms, we could start looking particularly at serotonergic GPCRs, or at there are 15 different trip channels in the worm. And doing those target-based screens, and perhaps somewhat surprisingly, we recently discovered a trip channel of the melastatin class, which we call Schistosoma mansoni TRPM praziquantel, that is activated by R-praziquantel. So interestingly, a sort of change in the stereoselectivity here between humans and worms, but at nanomolar concentrations. And we recently had a talented postdoc in the lab sort of who discovered and published that. So if you look at a bit of data, this is sort of calcium imaging data in HEC cells. Top is sort of the control cells. You can see when you don't put the flatworm ion channel in there, praziquantel does nothing, even though they respond to ATP. Whereas in the bottom three panels, when the cells are expressing the flatworm trip channel, you can see that R-praziquantel gives a quick and sustained and robust calcium signal, and at the same concentration, S-praziquantel doesn't do anything, even though the cells are still responsive. The stereoselectivity of that response, as well as the kinetics of that response, as you can see, look quite similar to what I showed you data earlier on the worm: nanomolar potency, stereoselectivity, large sustained calcium influx. So that was quite encouraging. So we have a potential target. It's a little correlative, saying, okay, this sort of looks like this, but it gave us a target to get our teeth into. And really, what we wanted to accumulate was more evidence that the properties of this channel reflected worm behavior. And I was pretty lucky. I was back in Liverpool at one of the Trop Med conferences just before the pandemic started. And Trop Med conferences, there are a lot of sort of NGOs, sort of companies here, and I got talking with Merck, and Jutta is sort of the head of Merck's Global Health Institute that's based in Geneva. And she was able to put me in contact with people at Merck that were still interested in this old drug that they discovered back in the 70s. And that was quite important because they were able to unlock their compound vaults and give us access to a whole series of praziquantel derivatives that we would never, as a lab, been able to synthesize. So they put in the mail, they sent blinded to us a set of 43 praziquantel derivatives, the enantiomers, metabolites, etc. And we were able, therefore, to quite simply screen these against our target. And we're able to find analogs that, like praziquantel, really sort of potently activated this trip channel, analogs that were a little less good, analogs that were sort of potent in the micromolar range, and just as importantly, analogs that didn't really work at all. So you can see through these slides, we could kind of put together a heat map characterizing the SAR of this flatworm trip channel. We could then go to work on the worms and do pretty much the same thing, put the same analogs on the worms and sort of color code which analogs had no effect at causing worm contraction, and for those that did, how potent they were able to contract the worms. And that sort of analysis could give us a sort of phenotypic heat map, and we can compare the phenotypic heat map with the characteristics of the channel, and you can see that they look pretty close. So we feel that the pharmacological fingerprint of this channel actually very closely reflects the ability of these same compounds to contract and ideally eliminate the worms from the body. There's actually a bit more data in here rather than just activity and contraction. There's also structure. So from this analog series, we're also able to put together a sort of ligand-based model. And what you can see under this color here is praziquantel. The cyclohexyl ring of praziquantel at this end is obscured by a region that is necessarily hydrophobic. So if you stick polar substituents on that ring, you're unable to activate the channel. And if you move down the praziquantel molecule past the carbonyls, there's another region that is predicted to be necessarily to be hydrophobic, to have hydrophobic substituents. And I'm going to come back to that later, because the ligand base inside basically says here are the characteristics of the ligand that you need to activate this channel. And then we can sort of compare this ultimately to how the key fits in the log. The characteristics of the channel, are they compatible with this ligand heat map? So this is what the worm channel looks like. It's a long trip channel. It has characteristic trip channel organization, the 6 TM domains and cytoplasmic regions. At the C-terminal end, it has this sort of green enzyme-like domain, which is typically found in human TRPM2 channels, regulated by ADP ribose. We also know from what I told you a little bit earlier that S-praziquantel at least can activate human TRPM8 channels. So we were interested in finding out, well, on the worm channel, where exactly does praziquantel bind? Is it like TRPM2, or is it binding to N and C terminal regions, or is it like TRPM8 and binding to the canonical transmembrane pocket that exists in the menthol receptor? So we were helped out a lot by the people at Merck, who were able to homology model the worm channel using known human crystal structures. Lucas is a very talented postdoc who predicted, and this is a model, that praziquantel was binding at the base of the voltage sensor-like domain in the worm trip channel, so at the base of transmembrane helices 1 to 4. And what we did is we sort of looked at all the residues in this binding pose that was close to the binding site of praziquantel, so within 5 angstroms. And this is shown here on this picture. And if we look at it a little bit more schematically and look at the characteristics of those residues, there was quite reasonable congruence with the ligand binding model. So there was an area of hydrophobicity around this cyclohexyl ring, residues that were quite hydrophobic, another area of hydrophobicity at the other end of the molecule. So it was good agreement with the ligand mapping. If we build up that binding site through sort of the 4 transmembrane helices and the trip domain, what I can show you is that cyclohexyl ring is actually surrounded by a whole suite of hydrophobic residues, phenylalanines, sort of tyrosine, leucine that sits on top of it. Similarly, at the other end of the molecule, again, this praziquantel seems to bridge to another hydrophobic pocket where there's this sort of tyrosine residue that forms nice pi-pi stacking with the ligand. And then in the middle, there's potential H-bond interactions from this arginine and this asparagine on the other side. So it's not a structure, but it's a model, but there was some good similarity there. We could test this model by mutagenesis. So we mutagenized all 23 residues that surround this binding pocket. I can just easily summarize that data. These were all alanine mutants, and we'll just color code them. So if a mutant ablated praziquantel activity, we'll put it as red. If it made praziquantel activation less sensitive in orange, and if that had no effect, green. What we found that if we screwed with this hydrophobic binding pocket at this end of the molecule, praziquantel no longer worked. Similarly, if we messed with this tyrosine or the residues around the other residues, also it didn't work. Mutation of the H bonding residues to the carbonyls were also poorly tolerated. So overall, I think we could mutate 20 out of 23 of these residues and interfere with the ability of praziquantel to activate the channel, which seemed reasonably good support for Lucas's binding model. So why is all of this important? Well, I think it's important because we can use this information to help us understand worm sensitivity to praziquantel. So if we take all those residues and think about that in terms of sort of a linear barcode, we've got 23 residues here. Let's go fishing to see in other trip channels, in other parasitic flatworms that are supposedly treated by praziquantel, whether they're sensitive or not. So first, you could look at the other schistosome species, and if you look bioinformatically, the predicted binding pocket residues are completely conserved in the other schisto species. And if you profile the channel functionally, this is control data, our Mansoni TRPM channel. And then if you look at either Hematobium or Japonicum, you can see that this channel robustly responds to treatment with praziquantel. You can go to those liver flukes, you can go to Clonorchis and Opisthochus. Again, that binding pocket residue seems to be completely conserved. And then if you functionally profile the channel, you can see those TRPM homologs in these other worms also respond to praziquantel. The one exception we had is, I mentioned at the start, is this liver fluke, Fasciola. So this was a key organism to try to look at. So we naively thought that Fasciola doesn't respond to praziquantel. Maybe this binding pocket is completely jumbled and praziquantel doesn't work. But bioinformatically, there just seemed to be one difference in this binding pocket, the substitution of a threonine for asparagine residue that exists in schisto. And that's quite a conservative substitution. Both these are sort of uncharged polar side chains, both can hydrogen bond. So we thought, aha, there goes our carefully crafted argument. This is not likely to be true, but we made some mutants. We tested this. Here's the schisto channel and the control again. And then when we looked at Fasciola, it was completely insensitive to praziquantel. So dead as a doornail. This channel didn't seem to respond at all. That amino acid variation, where was it? Where is this asparagine to threonine? If we go back to our model, it seems to sit as this residue that we predict interacts and holds that praziquantel carbonyl in place in the binding pocket. So at least it looks as though a residue that is in an important place. So let's do some mutants. Let's take the schisto channel and then let's make that substitution in the backbone of the schisto channel. And then when you did that, the schisto channel also failed to respond to praziquantel. That's sort of an easy experiment to do, make something that is working not work. Many of us achieve that every day in the laboratory. The cheekier experiment is, of course, the reciprocal mutation, is to take the Fasciola channel and substitute in the asparagine. And when you do it, take that channel, take the Fasciola hepatica channel, which doesn't respond to praziquantel, and you make the T2N mutant, you get a robust gain of function. And that channel now responds robustly to praziquantel. So we believe that we can explain why Fasciola is not treatable in people and in livestock in response to praziquantel by this natural amino acid variation in what we predict is the praziquantel binding pocket. So that was a quick whirlwind through our work on the channel. What I think hopefully I've explained is that we found the schistosome trip channel that responds to praziquantel with nanomolar sensitivity. We think pharmacologically the SAR of this channel really mirrors the action of praziquantel on the worms to contract them and to expel them. Homology modeling and mutagenesis seem to confirm that praziquantel is likely interacting in this transmembrane binding pocket in the worm TRPM channel. Homologs of this channel in other critters that we can treat with praziquantel also respond to praziquantel, and we can show that's because this binding pocket is conserved, and that's with the exception of Fasciola, which is caused by its insensitivity, we believe, is caused by this single amino acid variation. So I'm just going to end just 1 or 2 minutes just by saying about where we're going to go next. Our lab is really interested in drugs. We want to identify drugs to try and combat some of these diseases. And I think this, in terms of public health, is a particularly exciting time, because if we've just engaged in these mass drug administration campaigns, largely in sub-Saharan Africa, to try and swamp endemic areas with praziquantel. And for that to work, we've got to keep praziquantel working in the field. And if I just show you quickly a little bit of field data, this is a graph of schisto infections around Lake Victoria in Kenya over the course of, I think, 5 years of being prescribed praziquantel based on sort of a Gates campaign initiative. And you can see, if you sort of look around sort of the eastern shores of the lake, that these red dots are sort of rapidly disappearing as you go through time. So that's great. Praziquantel works in the field. We want that to continue. If you look in the area that's circled, what you can see is, well, there are some areas where sort of infection hotspots remain in the face of praziquantel treatment, and that there might be many reasons for that. But one reason we'd obviously be concerned about is if this is resistance being generated in the field to sustain selective pressure. We don't know that yet. We've not been able to sample worms from these hotspots, but we do know in the lab it's certainly possible to generate strains of schistosomes that are refractory to praziquantel. And I just quickly want to introduce data from Tim Anderson's lab, who is in Texas, who by treating snails with praziquantel, he can generate strains of schistosomes that are either praziquantel resistant or praziquantel sensitive. And through some beautiful sort of genetic mapping that Tim, Winker and Fred have been able to do, they've been able to do genomic association of variation that underpins this resistance traits. He's got associative peaks in the schistogenome, and actually under one of those peaks, if you look at the genes, the top hit gene for generating schistoresistance is actually this schisto TRPM channel that we've been looking at. So we're very keen Tim is very keen to look in the field to see if there are mutations in coding and non-coding regions of this channel that could lead to treatment failures in the field. Our lab, we're kind of interested in finding new drugs, so we're about to sort of launch a high throughput screen of the schisto channel. So we're partnering with Scripps down in Florida, and we're going to screen the collection against the channel. It's a bit of a fishing exercise, but hopefully it might come up with some new drugs. The pilot screen actually has come up with at least one new chemotype. So we have a new agonist that seems to activate this channel. It's a different chemotype, and actually on Tim's resistant schisto strains, it's able to convert the resistant strain to be more sensitive. So it must be working in the binding pocket somewhat differently. And then finally, something we'll be keen to do is, if we're right, this Fasciola disease that causes a lot of concerns about food security and loss of sort of meat production in livestock, is this due to this lack of praziquantel being able to be anchored into this binding pocket by this threonine variation. We know in the schistochannels and those that are sensitive, there's this asparagine residue there that seems to be crucial for anchoring praziquantel. So can we really design praziquantel derivatives that can better engage the natural threonine residue that's at that spot? So that gives you an overview of sort of what mischief we've been up to. I'd like to thank everyone in our lab who has been working on this, on host and parasite targets over the last actually decade probably. Also like to thank people at Merck who have really been quite helpful for giving us praziquantel analogs and helping us with modeling. And then also Tim's lab for helping us with looking at things in the field and scripts for helping us do starting to do drug screens. So I will shut up and I'll be happy to take any questions. Thank you.
Great, thank you, Jonathan. Fantastic talk, and very impressive results. So before I raise the questions in the chat box, I want to ask you a couple questions. So I'm an ion channel person. I'm very interested in this warm TRPM channels. I know in some ancient invertebrates, TRPM2 like channels is the only TRPM channel existing in those invertebrates. So is this warm TRPM it also the only TRPM channel in this worm?
So in terms of phylogeny, I think the TRPM subfamily is the most prevalent in the worm. So I think there are 6 or 7 TRPMs. It's kind of difficult to say what they're like because the homology is so low. I mean, this is a typical invertebrate problem. So there are 15 TRP channels in total. I think the only other, there are no TRPV channels predicted in parasitic flatworms. So that seems to be the only class that's missing. So I think likely those 15 TRPs have probably specialized along their own, unique routes. But I think it would be really fascinating to understand for all of them what their roles are in sensory physiology. I mean, obviously parasites are creatures that have plagued humanity for centuries, but if you think about their life cycle, it's really fascinating that the different environments that they have to persist through, right? I mean, the schistosome is in pond water, it's in human blood, it's in a snail, right? So in terms of signal transduction, thinking how an organism, one genome, how its stem cells reinvent itself into creatures that can exist and guide itself through all those different environments. So a long-winded answer. There are multiple TRPM channels. What exactly are their homologs in humans? I think we don't really know. And for those that have been characterized, their pharmacology also seems quite mixed up compared to the humans. So even though there are no TRPV channels, capsaicin still works on, for instance, some of the other flatworm TRPs.
Okay, thank you. So the NUDT nitromer, you have mentioned that in this warm TRPM2, does it have the maybe I should ask myself, but maybe you know the answer. Does it have the enzymatic activity like the human or the invertebrate TRPM2 that can hydrolyze ADP ribose into AMP and R5LP, or is this ADPR binding site conserved as in the human or invertebrate TRPM2?
Yeah, so I mean, we have no data to address that. We haven't really looked at the functionality of that domain. I mean, my understanding is it's also still maybe somewhat of a controversial issue in some of the human TRPMs. I don't think the channel is responsive to ADP ribose despite having this domain, so we can't get ADP ribose to activate this particular TRP. And the positive control for that is the Nematostella TRP, which does, in our hands, respond well. So we know that we're not seeing a non-response because of a lack of a positive control. It doesn't seem to be sensitive. It would it was one of our initial ideas that it might be sensitive because sensing redox might be an important thing for helping guide the worms through their life cycle stages, but it doesn't seem to work.
Okay, thank you. I will raise a question from the chat box. The first question is from Dr. Abril. Fantastic talk. My questions. The first question is, are there other unwanted effects of PZQ related to human trip channel modulation? The second question is, do you plan to confirm the binding pocket?
Oh, great questions. First one, unwanted effects of praziquantel on human trips. So S-praziquantel activates human TRPM8 with an EC50 of, as I showed in the talk, around 20 micromolar. I think there's been some interest in looking at it as a potential therapy, although the dosing is obviously very high in diseases where TRPM8 dysfunction has been implicated. Prostate cancer may be one of them. I think really the unwanted effect, to my mind, is could well be if the TRPM8 is mediating this bitter taste effect, which I'm not a taste physiologist, but I suspect it could. That's a huge effect about compliance in field medicine. So whether that I would love to do a clinical trial to see if just adding menthol to praziquantel ensured better uptake in the field. That could probably be quite effective. I not aware of any other negative effects. Do you plan cryo-EM to provide the binding pocket? That's something we would love to be able to do, because as I pointed out in the talk, even the modeling has been quite prescient. It is just not a model, not a structure.
The next question from Delia Alansare. Thanks, Jonathan. Very interesting talk. First, your essay showing the effect of PZQ on muscle contraction of the worms is very convincing. But how does PZQ affect the more pathogenic eggs? The second question is the worm TRPM activated by reactive oxygen species, or by cyclic ADPR like TRPM2, or by menthol like TRPM8? Do you want to answer the first two questions, then ask the third one? I'm afraid you may forget the first question. Read all three questions.
Sure. Yeah. So I think the idea in terms of anti-helminthic chemotherapy is to get rid of the worms, because if you get rid of the worms, then there are no eggs. So I mean, I think that we're largely targeting worm biology rather than targeting immunomodulatory things that would repress T cell responses to the eggs. So I don't think there's any data out there about how praziquantel affects development of larvae in the eggs. We're largely targeting the worm. I mean, it's a great question, because there is a huge amount of interest on what it is about these infections with parasitic flatworms that cause immune suppression in the body. And I think there's a lot of clinical data out there. I'm not going to say how robust it is, about people with autoimmune diseases, and I'm sure you've seen in the press about infecting themselves with flatworms. Tapeworms is the prime example, to quite effective at suppressing autoimmune symptoms. So immune modulation is a strategy, but largely for curing acute infections, we're going after the worms. As I said a little bit in response to the previous question, we haven't got any evidence. We don't know what the endogenous agonist is for this channel. If it seems to be a TRPM8-like, in human terms, if it seems to be a TRPM8-like channel masquerading in a TRPM2-like structure, that's just because of flatworm uniqueness. I think it's still a question. Someone else on the call can probably correct me, what occupies the binding pocket in human TRPM8. I think that's may still be unclear.
Okay, thank you. The third question is the warm TRPM channel possibly glycosylated critical asparagine residue.
Yeah, that good point. We haven't looked at that and perhaps we should. I mean, it's predicted to be inwardly facing into the pocket, but we should look at that.
Okay. The next question from Taufik Rahman. Great talk, Jonathan. Any idea why the prasequental fails to activate the Fasciola TRPM8 channel based on predictive modeling and docking? Is this isomer inherently unable to adopt the conformation needed to activate the channel? Many thanks.
Yeah, that's good. That's a great question. So I think looking at the structures of all the prasequantal analogs we screened, it seems that what is crucial is the orientation between the ring structures at the different ends of the molecule. So if you align, for instance, on the cyclohexyl and the carbonyls, the two enantiomers have different tilts on those ring structures. So it's a bit qualitative, but I think unless you have the correct orientation, you can't bridge those two regions of hydrophobicity, those two binding pockets. And that's also confirmed not just with the enantiomers, but also with other really small modifications, like epsiprantal is a 7-membered ring, and again it shows this tilt. So I think getting the both ends of that molecule correctly orientated is the reason why the enantiomer S prasequantal doesn't activate the worm channel.
The next question from John McCreel. Really nice project. Does the warm TRPM act as a cold sensing thermal receptor like vertebrate TRPM8? Is it agonized by methyl?
It's not activated by menthol. I think we screened a couple of typical human TRPM activators, and menthol and isolin and things don't activate it. Very good point about, you know, could there be another environmental cue that's activating it? In optimizing for high throughput screening, which we do at room temperature, we don't see any change in the EC50 for the channel, and we haven't taken temperature control any further than that, because I sort of think if you're an African schistosome, I wonder what temperature extreme you're actually exposed to beyond room temperature and 37. But I think John's right that, you know, there could be an environmental cue. Maybe there's certainly for some of the other trips, temperature regulation or mechano sensation or sensation of other things than redox. So, yep, I think that would be something that we would love to look at.
Okay. The next question is from Agnieszka Darda. Is TRPM expression stage depend in the flat box?
So that's a good question, and while it's a particularly important question, because prasequantal is notoriously ineffective at killing juvenile worms, so that when you treat a kid with prasequantal, it never quite, in many cases, eliminates the infection, because you've still got juvenile worms maturing in the body. And I think everyone sort of expected, oh, well, the answer to that problem is going to be because the target's not there. If you look at the expression of this channel across parasitic lifestyle stages, it's actually present in all life cycle stages, slightly upregulated and downregulated depending on what stage the parasite's at or what sex the parasite is. So that left us with a conundrum, is, you know, sort of explaining how juvenile worms are resistant to prasequantal if the target is still present. And I think probably the answer to that is actually in the ability of the worm to buffer the excitotoxic calcium damage. So I would say that I think the juvenile worms are packed full of stem cells because they're young worms, and as we can relate to, as you get older, your ability to repair yourself sort of declines. So I think even though the channel's there, and probably these juvenile worms are still contracting, they're much better at recovering because they've got neoblasts that can repair damaged tissue.
Okay, thank you. So the last question is from Sandy. Are there PZQ sensitive trips in non-worms?
Not that I'm aware of at that level of potency. So I think praziquantel action seems to be a flatworm specific thing. So tapeworms, flukes. Nematodes, praziquantel doesn't work on, so doesn't work on elegans. I don't think anyone has bothered trying other critters beyond those. So it seems to be very much a platyhelminth specific therapeutic.
Okay, thank you. Last question from me. Does this worm trip channel sensitive? Is it sensitive to calcium? Can it be activated by calcium?
Yeah, that's an obvious, very good question, and we don't know. We haven't got EFIS to the level, or I guess we've done a bit of mutagenesis of the predicted calcium binding sites, and they do kill the channel activity, but we haven't got EFIS going at a stage that we can do those dose response curves to look for calcium regulation. We can record some. I mean, I think we're pretty confident we can record single channel activity in nominally calcium free conditions, but we haven't studied it to the extent of scrutiny that I would like to give a definitive answer on that.
It sounds like a lot of work needs to be done. Yeah. Thank you. So so it seems like we don't have more questions. Thanks, Jonathan, for the wonderful talk, and thanks all the audience, and thanks, Melina, organizing this webinar.
Yep. Well, thank you for everyone for coming. Much appreciated. I hope everyone has a good summer.
There's one more question from Antonio Valabo. Are negative secondary side effects of PZQ in human?
Oh, yeah. So that's a good question, and I think the honest answer to that is we don't really know, because welcome to the world of tropical field medicine. That you're giving this drug to generally quite sick people who don't feel well. So there's not a really extensive literature on what the drug might do. I mean, it's labeled as side effects of sort of migraine, abdominal discomforts. I mean, I don't think they're severe, but they're probably related to the ability of the drug to engage 5-HT2B receptors, so vasoconstriction, vascular effects, and whatever TRPM8 may well do. But generally, many of these antiparasitics aren't well researched simply because of the diseases that they're used to treat.
Great, thank you. Thanks, everyone.